Hello and welcome to Nikolay's genetics lessons, and today I have prepared two multiple choice questions for you, and the first question is: which of the following characteristics of Taq polymerase make it useful for PCR process? And here's a variants to choose from. Let's start with the end of this list. It is exclusively moves from the 3' end direction to 5' end direction. Imagine that this is double-stranded DNA, and here we have replication bubble, and somewhere here we have primer, because polymerase need primer in order to make a new strand of the DNA. Without primer, it just cannot attach and make a new strand of the DNA. And all you have to know that new strand of the DNA would grow always from 5' end to 3' end. It always grow in only one direction. So this is wrong statement. You also have to understand that Taq polymerase is used in PCR reaction, polymerase chain reaction, but what I have shown you here is just a normal replication of double-stranded DNA, but the principles are the same. Taq polymerase or any other polymerase only can extend new strand of the DNA from 5' end to 3' end. Next variant C, it has regions that are complementary to the primers. If you think that Taq polymerase needs certain sequence which it recognizes in order to attach to this primer and extend new strand of the DNA, it's not necessary, because it can attach to any sequence. It just needs primer, but the sequence of the primer doesn't matter. So this is also wrong statement. Next statement, it binds more readily than other polymerases to helicase. So imagine double-stranded DNA. It actually looks like this. So we actually have our DNA which winded, and in order to unwind it, there is special enzyme is used, special protein which we call helicase, which unbinds this double-stranded DNA. And in order to prevent binding of these two strands of the DNA, there is another type of protein is used, which we call single-strand binding protein, which prevent these two complementary strands of the DNA bind again. But this is true only for normal cellular processes. In PCR, we don't have no helicase, no these single-strand binding proteins, because the temperature that we set would be as high, which doesn't allow these two strands of the DNA to bind again. So it is about very close to 100 degrees of Celsius. So as you see, instead of helicase, we just use temperature in PCR, so we don't have this protein, and these two proteins do not bind to each other in normal cellular process. So as you see, the only correct answer would be answer A. It is heat stable and can withstand the heating step of the PCR. And this is true because we took this enzyme from the extremophilic bacteria that grows in the hot springs in very high temperatures, and it is effective at the degrees where other enzymes, for example human enzymes, would denaturate and not going to work at all. So the name itself, Taq, stands for the DNA polymerase 1, named after the thermophilic eubacterial microorganism Thermus aquaticus, from which it was originally isolated in 1976. And second question is very easy: which of the following statements about human reproduction is true? Variant A, mitosis in males is also known as spermatogenesis. It is false statement, because spermatogenesis or production of male gametes would be a process of meiosis, not mitosis. Variant B, sperm and ova are zygotes. No, zygotes would be fertilized egg cell, and sperm and ova would be gametes. So as you see, the only correct variant would be variant C, or genesis takes place in the ovaries of females, and this is true. If in males spermatogenesis is a process of production of new sperm cells, in females oogenesis is more a process of egg cell maturation. And this is all for today. Subscribe and see you in the next video. Goodbye.